I want you to just imagine for a second a sprawling, highly automated biological manufacturing line. Millions of tiny microscopic components are just zipping down a conveyor belt, being assembled, packaged, and shipped out.
Right, a massive scale.
Exactly, massive scale. But somewhere in an incredibly complex process, microscopic errors are happening. And these errors aren't just invisible, they're actually leaving behind a really distinct chemical fingerprint on the final product.
And that's a fingerprint our own immune system is, well, it's anciently hardwired to detect, flag, and attack.
Yeah, which is exactly why we are here. Welcome to the Deep Dive. Today, we're taking a close look at a recent preprint by researcher Kevin McKernan. We're going to examine the exact chemical fingerprints left behind on some mRNA vaccines, specifically looking at a lot from Pfizer.
And for some context here, McKernan is the researcher who initially broke the news about residual bacterial plasmid DNA in these vaccines.
Right, findings that actually passed the rigorous threshold of peer review later on.
Yeah, exactly. So because his previous work on this specific, biological contamination passed that peer review process, new data from his lab, even if it's in preprint form, it commands immediate attention. I mean, it really gives us an opportunity to look under the hood at the raw data and the actual molecular mechanics he's outlining.
Absolutely. Now, before we get into the molecular weeds here, it's really worth noting how we're approaching this today. Because our goal isn't to debate vaccine policy or politics.
No, not at all.
We are strictly looking at the raw biological data McKernan published. We're reading the exact genetic barcodes and understanding the molecular engineering so you, the listener, can understand the mechanics for yourself. We're essentially treating this like a microscopic detective story.
I like that, a detective story. Because the researchers use some highly advanced genetic sequencing technology to read the precise contents of what actually ended up inside these specific vaccine vials.
Right. And to understand the first major piece of evidence they uncovered, we kind of have to transition from the macro level of liquid vial down to the epigenetic layer of the DNA.
Yeah, epigenetics. It's basically the layer of chemical information that sits on top of the underlying A, C, T, G code.
So it doesn't change the sequence of the letters themselves?
No, not at all. But it absolutely changes how those letters are read, interpreted, and managed by a cell.
Okay, let's talk about the specific tool the investigating team used to read that epigenetic layer. Because it seems really foundational to how they even found this in the first place. They used an Oxford nanopore sequencing machine.
Yeah, the nanopore.
How does that actually work differently than older sequencing methods?
Well, older sequencing methods essentially rely on destruction. They take a long strand of DNA, chemically chop it up into tiny, manageable pieces, read those short fragments, and then use complex computer algorithms to guess how they all puzzle back together.
Oh, wow. So they're just guessing based on a bunch of tiny fragments.
Exactly. But the Oxford nanopore technology doesn't do that. It reads massive, perfectly intact genetic molecules from beginning to end. It physically threads the entire intact DNA strand through a microscopic protein pore that's embedded in a membrane.
Wait, it physically pulls the whole strand through.
Yeah. And as the strand moves through, it disrupts an electrical current. The machine reads that specific electrical signal in real time to identify each base pair. And crucially, this is the big part, it can simultaneously read the epigenetic chemical markers attached to those base pairs.
Okay, so it's not looking at a shredded document and guessing. It's literally reading the whole scroll.
Perfectly said, reading the whole scroll.
And when they ran this specific Pfizer vaccine sample through the Oxford nanopore sequencer, the sheer volume of what they found was substantial. I mean, they detected over 590,000 fragments of bacterial plasmid DNA in a single run.
Which is a huge number. And while the presence of DNA was the subject of McKernan's prior work, this new paper really zeroes in on the epigenetics, specifically a process called methylation.
Methylation. Okay, what exactly is that?
Methylation is just a fundamental biological mechanism. To put it simply, an organism attaches a tiny chemical, a methyl group, onto a specific letter of the DNA. Organisms use these tags for a variety of reasons, but a primary one is recognizing what is self and what is foreign.
Okay, I'm a bit hung up on this part. If human cells and bacterial cells both use these methyl tags, how does our immune system know the difference? Is it looking at the tag itself, or where the tag is placed?
It is entirely about where the tag is placed. Bacteria utilize specific enzymes, they're referred to as DAM enzymes in the literature, to methylate the nucleotide adenosine.
So they put the tag on the A.
Right. They scan the DNA for a very specific sequence of letters, and when they find it, they just slap a methyl tag right onto the adenosine. Humans do not do this. Our cellular machinery generally methylates a completely different pattern, specifically a cytosine followed by a guanine, which is known as a CG motif.
Okay, so the human cellular machinery expects to see tags on CG motifs, and it is definitely not expecting to see tags sitting on adenosine.
Exactly. And this brings us to the core consequence outlined in the research. When the mRNA vaccine is manufactured, it utilizes lipid nanoparticles.
Right, the microscopic fat bubbles.
Yeah, the fat bubbles. Their entire job is to protect the fragile genetic payload and deliver it safely across the human cell membrane into the interior of the cell. Normally, the human body would violently destroy free-floating genetic material in the bloodstream. The fat bubble acts as a stealth delivery vehicle.
But the fat bubbles aren't just packaging the intended mRNA, right? They're also packaging these residual fragments of bacterial plasmid DNA that were left over from the manufacturing process.
Unfortunately, yes.
And because that DNA was grown inside bacterial factories, it's covered head to toe in bacterial methylation tags. It's essentially like someone sneaking into a rival team's stadium wearing their home team jersey.
That's a great analogy.
I mean, the lipid nanoparticle sneaks them in the door, but once they're inside, they're wearing the completely wrong uniform.
Right. And the security guards in this scenario are complex sensor proteins located inside the cytoplasm of the human cell. When these sensors detect that foreign adenosine methylation pattern or, notice the stark lack of expected human CG methylation, they don't just quietly clean up the debris.
What do?
They activate interferon pathways.
Ah.
Yeah. Interferons are signaling proteins that effectively shout to the rest of the body that a viral or bacterial attack is underway. This triggers a massive cascade of cellular defenses and causes immediate inflammation. The body is reacting exactly the way evolutionary biology trained it to react when it detects a pathogen's fingerprint.
That makes total sense. But wait, if the bacteria is slapping methyl tags all over this plasmid to protect it or manage it, why does the data show that one specific section, the SV40 promoter, was completely unmethylated? I mean, that can't be a random accident, right?
No, it's highly unlikely to be an accident, and it highlights a fascinating aspect of molecular biology. So a promoter is a sequence of DNA that essentially acts as the biological on switch for reading a gene. The SV40 promoter, which is derived from a virus, is a remarkably strong, well-known on switch used heavily in genetic engineering. Now, in biological systems, heavy methylation often acts to silence a gene. The bulky methyl tags physically obstruct the cellular machinery that tries to bind to the DNA and read it.
Oh, I see. So by keeping the methyl tags off the SV40 promoter, the bacteria ensures that the on switch remains completely physically unencumbered. It stays highly active.
The data strongly suggests exactly this mechanism. You're left with foreign DNA that is heavily methylated in areas that produce an inflammatory immune response, but completely unmethylated at the exact regulatory site needed to drive continuous biological activity.
That is wild. But wait, if these fragments are so heavily methylated and wearing this glaring bacterial uniform, they shouldn't even be surviving in the vial in this state, right?
I think so.
Because the whole point of the manufacturing process is to chop this DNA up into harmless, unrecognizable debris.
Okay.
So how did massive, intact chunks of it make it into the final fat bubbles?
Well, to understand this mechanical failure, we have to look at how mRNA is manufactured at a massive scale. You use bacteria as tiny factories. You insert a circular piece of DNA, a plasmid, into the bacteria.
Okay, the plasmid.
Right. That plasmid contains the genetic instructions you want. The bacteria then naturally copy this circular DNA millions of times as they multiply.
But a finished mRNA vaccine isn't supposed to contain circular bacterial DNA.
No, absolutely not. Because circular DNA is incredibly durable. It is designed by nature to persist in harsh biological environments. So to manufacture the mRNA, the laboratory has to extract these circular plasmids from the bacteria and chemically cut them open so they form a straight line.
Okay, so making it linear.
Exactly. The process is called linearization.
Yeah.
Once it's a straight line, it serves as the template to print the mRNA. And after the mRNA is printed, that linear DNA template is supposed to be chopped up into tiny fragments and completely filtered away.
Got it. So if the manufacturing process uses precise molecular scissors to cut the circle, the Oxford nanopore data implies the scissors just missed the cut site entirely.
That is exactly what the sequencing read reveals. Because the nanopore sequencer can read massive, intact molecules without breaking them, it didn't just find random, tiny fragments of DNA. It read the genetic sequence right through the specific site where the chemical scissors were supposed to have made the cut.
Oh, yeah.
Yeah, the cut site was completely intact, proving the linearization process failed for a significant portion of the plasmids.
And the scale of these uncut fragments is staggering when you look at the preprint. The entire bacterial plasmid used to develop this specific construct is about 700-800 base pairs long.
Roughly, yeah.
But the sequencer was pulling out intact fragments that were up to 3,400 base pairs long. That is nearly half of the entire original circle surviving the process.
It's a massive, and that scale introduces a very specific, well-documented biological hazard. See, when DNA is chopped up into tiny pieces, maybe a few dozen base pairs long, it's essentially harmless cellular debris. The cell's waste disposal mechanisms clear it out easily. But when you have massive, contiguous fragments of a plasmid that were never properly linearized, you run into the severe problem of sticky ends.
Okay, let's get into the actual chemistry of sticky ends. DNA naturally wants to pair up, right? Adenine bonds with thymine, cytosine bonds with guanine.
Precisely. When DNA is broken randomly or cut improperly by certain enzymes, the two strands of the double helix don't break perfectly evenly. It leaves an overhang, a single strand of unpaired genetic letters dangling at the very end of the fragment.
Like a cliffhanger.
Yeah, and because of the basic laws of chemistry, specifically hydrogen bonding, these exposed, unpaired letters aggressively seek out their missing partners. They are chemically sticky.
So instead of a broken paper shredder, it's more like strips of Velcro.
That's a perfect way to visualize it.
The unpaired letters are the hook side of the Velcro aggressively seeking the loop side to bond with. So if you have massive fragments of a plasmid floating around inside a human cell with these exposed Velcro ends, they could just find each other and snap back together.
In molecular biology, this is called religation. If the sticky ends of a massive fragment or multiple fragments find complementary matches, they chemically bond back together. They can easily reform into full, intact, circular plasmids right inside the biological environment of the human body.
And as we established earlier, a circular plasmid is far more durable than a linear fragment. It violently resists being broken down by the cell's natural waste disposal system.
Exactly. A reformed plasmid can persist in a cellular environment for a long time. If that reformed, stable plasmid happens to contain the highly active SV40 promoter sequence we discussed earlier.
On switch.
Right, the on switch. It could theoretically continue to function. It could produce unintended genetic transcripts, completely independent of the mRNA payload the vaccine was supposed to deliver. And all while the bacterial methylation uniform continues to trigger a localized inflammatory response.
Okay, finding fully active, massive chunks of bacterial DNA that can basically Velcro themselves back together inside the human body.
Yeah.
That sounds like a catastrophic engineering failure.
It sounds alarming, for sure.
But reading through the preprint, the tone actually shifts dramatically in the final sections. This data doesn't just point out a problem, does it?
No, it doesn't.
It provides the exact technical coordinates for fixing the process. It shifts the focus entirely to the future of biological manufacturing.
That's the beauty of it. Identifying the exact molecular mechanism of a failure allows bioengineers to design a targeted, permanent solution. The preprint details several straightforward technical adjustments to ensure these specific flaws are engineered out of future iterations of mRNA technology.
Okay, let's look at the epigenetics first. The bacterial uniform causing the inflammation. How do you stop the bacteria from dressing the DNA in those methyl tags?
The solution relies on foundational genetic engineering. We can modify the specific strain of bacteria used in the factory phase so they simply lack the genes to produce DAM enzymes.
Oh, brilliant.
Right. If you knock out the bacteria's ability to manufacture the enzyme, it becomes physically impossible for them to attach the methyl tags to the adenosine nucleotides.
So if they can't make the uniform, they can't wear it. Even if a fragment of DNA accidentally makes it through the entire purification process and ends up in the final lipid nanoparticle, it will be naked.
Exactly. It won't have the bacterial methylation pattern to trip the human immune system's epigenetic alarms. It effectively neutralizes that specific inflammation trigger.
That is so elegant. Okay, what about the second proposed solution? Because that addresses the linearization problem, the Velcro.
Right. The authors outline how the restriction enzymes, the chemical scissors used to cut the circular plasmids, can be swapped out to completely eliminate the creation of sticky ends.
How does a different enzyme change the chemistry of the cut?
Well, you use a class of restriction enzymes that create what are called blunt ends. Instead of leaving a jagged overhang of unpaired letters looking for a partner, a blunt end enzyme cuts straight across both strands of the DNA double helix cleanly and evenly.
So no overhang at all.
None. There is no exposed hydrogen bonding potential.
It's like cutting a document and instantly laminating the edges. Without the hook and loop of the Velcro exposed, the fragments have a significantly lower probability of ever being able to re-eligate and form a circle, even if massive fragments somehow survive the shredding process.
That's exactly right. And the third solution addresses the survival of those fragments directly. It advocates for the implementation of far superior degradation and filtration systems at the factory level.
I mean, this seems like the most obvious step. Just clean the biological soup better before you package it.
It sounds simple, but it's critical. The current protocols rely on enzymes that are supposed to chew up the DNA template after the mRNA is printed. But as the Oxford Nanopore data proves, these enzymes are either failing to cut entirely or they're leaving massive chunks behind.
Right.
By upgrading to highly aggressive, redundant DNA-degrading enzymes and coupling that with advanced physical filtration techniques, the bacterial DNA can be utterly obliterated. This purification has to happen to an extreme standard before the mRNA is ever mixed with the lipid nanoparticles.
Because if the DNA contamination is eradicated from the vat, it can never get packaged into the delivery bubble in the first place.
Exactly.
Since this was the very first mass-scale rollout of mRNA vaccine technology in human history, catching these exact flaws feels like a really necessary, if uncomfortable, part of how complex biological engineering is supposed to iterate.
It is. The history of any novel medical technology, from the first polio vaccines to early recombinant proteins, shows that the first generation always reveals unforeseen manufacturing vulnerabilities when scaled up to billions of doses.
Sure.
What makes this current moment unique is the sheer power of our diagnostic tools. Oxford Nanopore sequencing allows us to catch a molecular error at the atomic level. This visibility allows the engineering process to adapt and improve at a speed that was impossible even a decade ago.
Let's synthesize all of this so we can see the complete picture here. Through the lens of advanced genetic sequencing, researchers discovered that microscopic errors in the manufacturing process left large bacterial DNA fragments in some mRNA vaccines. These fragments carried a bacterial epigenetic uniform, specifically adenosine methylation, that acts as a glaring trigger for human immune responses. Furthermore, the linearization process failed, leaving massive pieces of DNA with sticky Velcro-like ends that carry the biological risk of reforming into stable, highly durable circular plasmids right inside human cells.
Which is a lot to process.
It is. But crucially, mapping these exact flaws provides a highly specific technical blueprint. By genetically knocking out the bacteria's ability to methylate, ensuring molecular scissors make blunt cuts, and drastically improving filtration, the manufacturing process can be made incredibly clean.
The raw data demystifies the biological hazard and translates it directly into an engineering challenge with known, actionable solutions.
Which brings us back to you, the listener. Why should you care about the microscopic methylation of a cytosine versus an adenosine? Because understanding the actual physical mechanics of what goes into your body takes science out of the realm of magic or fear. It places it firmly into the realm of mechanics and engineering. And when you understand the engineering, it empowers you to expect and demand safer, highly refined iterations of these new medical technologies in the future. Knowledge gives you the leverage to hold the manufacturing process accountable to the highest possible biological standard.
It fundamentally changes the conversation,
Yeah.
from trusting a process blindly to verifying its precision at the molecular level.
Exactly. And I want to leave you with a final lingering question to ponder on your own. We've just spent this deep dive exploring how incredibly sensitive our genetic sequencing technology has become. We can catch microscopic epigenetic manufacturing errors long after the vials have been shipped, distributed, and administered.
Long after.
Right. So if our sequencing technology is now this undeniably powerful at playing detective after the fact, how might this radically change the way regulatory agencies monitor, sequence, and proactively audit all biological therapeutics before they ever leave the factory line? Think back to that biological manufacturing line we talked about at the start.
Moving at lightning speed.
The conveyor belt is moving faster than ever. But now we have a magnifying glass powerful enough to read the chemical fingerprints before the box is even sealed. Thanks for joining us on this deep dive. We'll see you next time.